Hello students, welcome to Zoology Classroom. In this video, let us know about biotechnology in livestock improvement. So in the previous video, we have seen about the scope and importance of biotechnology, and later we have seen several ART, assisted reproductive therapies. And in this video, let us know how this biotechnology is helping in livestock field. So the aim of biotechnology in the improvement of livestock health is to increase the genetic variations in the livestock and to increase the accuracy of selection, to reduce the generation intervals, and to increase the selection intensity. These are the four aims of biotechnology usage in livestock improvement. And to achieve these four goals or four aims, there are two ways. And they are, one is by manipulating the genes of the livestock. And second method is without manipulating the genes. First of all, let us see without gene manipulation methods. So there are seven methods that fall under this without gene manipulation method. First one is artificial insemination. Second one is multiple ovulation and embryo transfer technique. Third one is oocyte harvesting. Fourth one, in vitro oocyte maturation. Next, in vitro fertilization. Sixth one is nuclear transfer and embryo cloning. Seventh one is sex selection. Let us see about the first one, artificial insemination. So since the development of these artificial insemination method, first of all, freezing methods were discovered. So this efficient semen freezing methods to store the semen for a long time. So with the development of these efficient semen freezing methods, artificial insemination has a most widespread biotechnological method in livestock field, especially in cattle production. So by allowing for the widespread use of a small number of elite sites, so a very good quality sites, especially this bulls, they are very less in number. So to use the best quality semen from these elite sites or best quality bulls, artificial insemination has a better impact on the selection intensity. So in addition to this, this artificial insemination method has allowed for the implementation of the progeny testing scheme prevalent particularly in dairy cattle production. So before the dairy cattle are produced, progeny testing will be done. So this progeny testing method will be made possible with the help of this artificial insemination. And this method has a major impact on the improvement of herd by increasing the accuracy of selection despite the associated increase in generation interval. So artificial insemination also helps in the increase in generation interval. And apart from that, it is also helping in increasing the accuracy of selection qualities or selection traits. And second method is MOET, multiple ovulation and embryo transfer. So by increasing the number of offspring that can be obtained from monococcus species, in particular, so in these monococcus species, this MOET technology has a lot of potential to increase genetic improvement and to increase the selection intensity as well on the female side. So usually these monococcus species, they will give birth to only one baby at a time. So in such species, this multiple ovulation and embryo transfer method enables us to produce a good quality offspring from a good quality mother in large numbers. So normally a good quality mother or whatnot. So the female, they can give birth to only one baby, but with the help of this MOET, we can produce as many number of good quality offspring as we wish. So in cattle, this MOET technology is most widely used one, and it has a major impact on the in this cattle that it reduces the generation interval a lot. So generation interval reduction will be achieved with the help of this MOET. So there are many problems in doing this multiple ovulation and embryo transfer method, even though this MOET has a potential to play an important role in developing countries, especially in these developing countries. Implementation of a large-scale artificial insemination based on progeny testing scheme would be difficult to implement. So artificial insemination requires progeny testing. So if we want to conduct this artificial insemination on a large scale, progeny testing scheme will become difficult. So in such cases, MOET is very, very useful in developing countries. So what is the procedure that is followed in this MOET? We can see in this slide. So first, super ovulation will be done from in a donor cow, and some gonadotrophins will be injected to it, so that with the help of these gonadotrophins, it will produce a lot of eggs at the same time. So after this injection of gonadotrophins, after 5 days, artificial insemination will be done. Artificially, they will introduce semen into the uterus of this cow, and 6 to 8 days later, embryos, fertilized embryos will be collected from this cow. And for the collection, to collect the embryos, the Foley catheter will be used. So after collecting them, they will be examined under the microscope whether they are properly developing or not. So, and based on the microscopical observations, they are classified. So these classified very good quality embryos, they are preserved with the help of liquid nitrogen at minus degrees, or for storing for just 1 day. Storing at 37-degree temperature is enough. But if we want to store them indefinitely, then we have to go for very sub-zero temperatures. So later, through surgical methods or through non-surgical methods, these embryos can be transferred into the recipient cow's uteri. So, and the development of these embryos in the recipient's uterus will be done with the help of palpation method. In this palpation method, the examiner puts his hand through the rectal wall, and up to 1 to 3 months of baby's embryo development can be seen by this palpation method. So if everything goes well, after 9 months, this recipient will give birth to a baby. So likewise, all the collected embryos from this donor, each one egg will be introduced into a different recipient. Like this, good quality embryos can be collected from this superior donors, and these can be transferred into ordinary recipients, so that these are ordinary recipients, they are able to give birth to a good quality offspring. So this is the procedure methods are followed in MOET technique. So, and next one is oocyte harvestation. This is also called as ovum pickup method, OPU method. And this OPU method will always be coupled with in vitro oocyte maturation, IVM, and in vitro fertilization, IVF methods. So both these three methods are collectively used to achieve the required or desired results. So in this oocyte harvesting or ovum pickup method, what they will do. So in normal conditions, MOET is not possible. So for example, when the good quality donor is on gestation period, then we cannot, normally we cannot procure oocytes from the uterus of that donor. So in this case, what we can do, we can perform a surgery like keyhole surgeries or laparoscopic surgeries, and directly we can collect oocytes from the ovaries of those good quality donors. So like this, by operating or by doing a minor surgery, buttonhole surgery, we can collect as many as embryos as we require or we want from the ovaries of those organisms. So this is called oocyte harvesting. So directly we are collecting oocytes from the ovaries. In MOET method, what they will do, they will collect the ova from the uterus or from the fallopian tubes. But here in this oocyte harvestation, before the oocytes are released from the follicles, okay, before the oocytes are released from the follicles, they will be directly collected from the ovaries. So, and after collection of oocytes or mother oocyte cells from the ovaries is called oocyte harvesting or ovum pickup. So after the pickup of these ovum from the ovaries, they will be subjected to in vitro oocyte maturation. So in oocyte or in ovum pickup, the ova are not matured. So they did not complete the meiosis 1 and meiosis 2 phases. So the maturation is not done in normal conditions. So before they are matured, we have collected them. So now we have to allow them to mature in vitro conditions. So in laboratories, we will allow them to mature. This is called in vitro oocyte maturation. So after the ova are matured in vitro, then we will perform this in vitro fertilization. So we will take the semen and we will mix these matured ova and the semen. So in vitro fertilization results in the development of embryo. So these developing embryos, again, they will be projected, or they will be kept in the uteri of the recipient. So in this way, oocyte harvesting or OPU, OVM, IVM and IVF, these three methods can be used instead of MOET method in some cases. So here in this method, from this superior donor, before the oocytes are released, so in MOET, we will give gonadotropins so that superovulation is achieved. So here in this case, we will not give gonadotropins. So directly we will do some minor surgery and we will collect immature oocytes from the ovaries. So these oocytes are allowed to mature in vitro, and fertilization also will be done in vitro conditions. So these developing embryos, they are introduced into the uteri of recipients. So this is happens in this ovum pickup and in vitro ovum maturation and in vitro fertilization methods. And next one is nuclear transfer or embryo cloning. So nucleotransfer method involves the transfer. This is nothing but normal, this what Dali production method or cloning. So this is just like animal cloning. So in this thing, totipotent nuclei will be collected from oocysts, from not only oocysts, from any other cell of a superior donor mother. So from superior donor mother or superior donor father, or it may be any animal. So we can collect the nuclei either from male organisms or female organisms, but that organism should be having required qualities. So we will take the totipotent nuclei from that recipient nuclei donor. And then later we will collect the eggs, oocytes of the mother, and these oocytes are anucleated. So nuclei will be removed from this oocyte. So now the oocyte is having just ooplasm. So into this enucleated oocytes, we can introduce these totipotent nuclei. So and this results in the artificial formation of zygote. So these zygotes, again, they are introduced into the uterus of recipients. In the recipient uteri, they will develop normally, and the recipient gives birth to the baby after the gestation period is over. So through this method, what will happen? The baby or the offspring which has taken birth will be similar to the nuclei donor. So with this method, we have got this dolly animal for the first time. So nucleotransfer is nothing but producing cloning animals. So this is the organism to be cloned. We will collect eggs from this, cells from this thing. So this organism is having superior quality. So we want to have a clone to this organism. So similar type of organism is required for us. So we can collect cells from any other organ of this donor. So in case of Dolly, they have taken this cells from this mammary gland. So these cells are giving nuclei. So they are nuclei donor. And from some other animal, ova will be taken. This ova will be enucleated. Nucleus will be removed from them. So into this enucleated ova, this nucleus will be added, so that an artificial embryo or an artificial zygote will be formed. This zygote starts developing. So later, the early stage embryos, they will be transferred into this recipient, and in this recipient, after the gestation period is over, then what will happen? The born organism, it will be similar in all aspects except in size and except in age. In all aspects, it will be similar to this nuclear donor. So this is the method that will be followed in nuclear transfer method. And last one in this category, first category, is sex selection. So this is nothing but sperm sexing. So we have also seen the procedure of sperm sexing in previous video. So in this thing, we can select the required sperms, whether the X chromosome bearing sperms or Y chromosome bearing sperms. So depending on the requirements, if we want to have a female offspring, we can have X chromosome. We can separate X chromosome sperms only from the semen, and we will use these X sperms in artificial in vitro fertilization techniques. So if we want to have only male offspring, so these male offspring usually are required in meat producing industries, and female offspring are required in dairy industries. So different industries are having a different requirement. So depending on the requirement, either they will go for X sperms or they will go for Y sperms. So sperm sexing technologies also have been developed, and they are widely in use. And after the separation of sperms, depending on their sex chromosomes, the selected sperms will be used in artificial insemination and artificial in vitro fertilization methods. Later again, they will be used in embryo transfer methods. So apart from this sperm sexing, embryo sexing also can be achieved by microbiopsy and sex determination using polymerase chain reaction amplified by Y-specific sequences. So we can also determine, detect the sex of the developing embryo by using this PCR test. In this PCR test, to determine the sex of the developing embryo, some X-specific sequence DNA parts are tested, whether they are present in the embryo or not. If the embryo is having Y-specific sequences, then we can conclude that the developing embryo is a male offspring. If the Y-specific sequences are absent in the embryo, developing embryo, then we can know that it is a female offspring. So in this way, embryo sexing also is possible. So this approach, embryo sexing, is economically not viable. It is only justified in only very, very rare exceptional cases. Next, coming to the genetical modification methods in the, so before that we will move on to genetical modification methods. There is another method also there which does not involve the gene manipulation methods, that is gamete and embryo cryopreservation. So this cryopreservation method also we have seen in detail in another video. So through this cryopreservation, we can store the gametes, we can store the embryos at sub-zero temperatures for a long period of time. Say, for example, say for several years also we can store them. So this cryopreservation also helps a lot in this cattle livestock production procedures. So with the help of this cryopreservation also, we can store the good quality sperms and good quality embryos for a long time. So that this method also is helping us in maintaining genetic diversity. Next, coming to moving on to genetically modification methods. So in this genetically modification methods, we require this rDNA technology methods mostly. So advancements in the molecular genetics fields have boosted to this human genome project. So with the help of this human genome project, we were able to know some genetic markers inside our genome. So these genetic markers are very, very useful in determining the location of the genes and presence of the genes. If the markers are there, we can know the gene is there. If the markers are absent, then we can know the genes are not there. So in this way, to determine, to identify the genes, to identify the working of the genes, genetic markers are very, very important. So for the first time, this human genome project has led us to know the genetic markers in humans. So this technology or these methods have also been used in this livestock health development practices also. So this is a fundamental tool of the geneticist. So these markers can be used to locate the genes, and these genes are very, very important in creating or in exhibiting phenotypic traits. So traits are very, very important in case of livestock. Milk production is a trait, meat production is a trait, and giving milk in very less time is also a trait. So like this, different traits are there. So all these traits are directly are dependent on genes. So identifying, locating the genes is very, very important. So for this purpose, genetic markers are very useful. And these genetic markers are also useful in genome map, in constructing genome maps. Construction of genome maps uses a linkage experiments. So linkage experiments also help in production of construction of genome maps. In this case also, scientists will depend on genetic markers. So this genetic mapping is the first step in the process of positional cloning. This positional cloning culminates the isolation of the causal gene and mutation. So if a gene is causing a very undesirable trait in the organisms, and if a gene is causing some dangerous mutations, then we can culminate it, we can remove that gene. So in this way also, to identify such undesirable genes are also, this mapping will be very useful. So understanding the molecular biology of production of traits is also very, very important in all these experiments. So it is strongly believed that the identification of QTL will allow for the implementation of novel marker assisted selection schemes. So QTL is one type of method used in this genetic mapping. So this identification of QTL sequences helps in developing marker assisted selection methods. So these marker assisted selection methods are expected to increase the genetic response by affecting all the 4 relevant factors that we are intended to increase with this biotechnological methods in the improvement of livestock health. So mapping of genes explains breeding differences for economically important traits. And this also will allow their introgression in other populations by marker aided backcrossing. So therefore increasing the genetic variation usable acerbate for selection programs. So, and in genetic engineering also, with the help of this rDNA technology, we are able to produce different genetically modified organisms. So several genetically modified organisms also have been developed in livestock also. So the purpose of these genetic engineering methods is to have a recipient organism take on the characteristic controlled by the transferred gene. So in this way, so far with the help of the genetic engineering method, not very good number of genetically modified organisms have been produced in case of these livestock head, but in other organisms they have been produced in a large number of instances. So in future we can expect this genetic engineering method helps a lot in the improvement of livestock head. So and now another best use of gene manipulation in livestock head is BST hormone. So BST is a hormone. It is called as a bovine somatotrophin. So this is also known as bovine growth hormone. So this bovine somatotrophin, this helps in the growth of the organism, growth of the livestock animal. So somatotrophin hormones, they will affect the utilization of energy in the body. And these hormones are these BST causes energy derived from feed to be used for milk production rather than weight gain. So previously, the only source of VST for research has been from pituitary glands of dead cows. But now with the help of rDNA technology, genetic engineering methods, large quantities of BST can be produced in microorganisms also with the help of rDNA technology. And future challenges in this rDNA technology or gene manipulations is increasing production efficiency for animal products, either by manipulation of the genes or manipulation of the environment, remains a major component of the available means to achieve proper feeding of the growing world population in a sustainable way. So this is the major challenge. And biotechnology is an essential part of the arsenal of tools needed to reach this objective. And as the methods continue to develop, the contribution of the biotechnology also is likely to increase in the future. And major problems related to the applications of biotechnology in animal production as identified by a growing distrust in and fear of biotechnology by the general public. So general public are not trusting, so distrust is being growing day by day, and some people are also having fear to use the products of biotechnology, like GM food are not they are a subject of controversy. So some people are advocating for them, and some other people are against the use of genetically modified food. So they are arguing that these GM food, they will damage our health. So in this way, some distrust is being growing, and some fear is also being growing in general public. So this is one major problem. So scarcity of properly educated scientists, technicians, and lack of support for the projects also a major problem in this biotechnological field of improving animal livestock heads. So doubts about the cost effectiveness of biotechnology applied to animal production also is there. So these are the three major problems in this livestock improvement by biotechnological methods. So with this topic is over. Let us meet with other topic in next class.